Hi everyone, and thanks for joining us at HCLS Web Day. My name is Erin, and I'm the Life Sciences Lead on the Open Data team. Today, I'm going to focus on open genomics data because we believe our genomics customers are using it to change the world. On the AWS Open Data team, we help communities grow around open data. People who use open data often build new tools and products that make all data easier to work with. Open genomics datasets listed on the Registry of Open Data range from cancer genomic datasets to genome assemblies of endangered species to clinical annotations of genetic variants. So if you're looking to access data that you know lives in the public domain, please visit the Registry of Open Data or email us at opendata@amazon.com. We're here to make sure you have access to the data that you need. Open data has never played a more important role in society than it does today in the midst of a global pandemic. As Nobel laureate Dr. Jennifer Doudna said in an opinion piece to The Economist, "We're releasing data as soon as we have it. We're collaborating across disciplines and time zones, and the public is counting on us. A new era of science is emerging. It can't arrive soon enough." And perhaps there is no other genomic dataset that embodies a new open era of science like the Genome Aggregation Database, or gnomAD. gnomAD is the largest publicly available catalog of naturally occurring human genetic variation in the world. Its most recent release catalogs over 640 million genetic variants annotated from 71,702 whole genome sequences. Today, we have Grace Tiao of the Broad Institute to tell us more about gnomAD, its inception and intent, and how the cloud has been intrinsic to its genesis and its distribution to the world. Grace, thank you for joining us.
Thank you, Erin. I'm so thrilled to have the opportunity to share with you all today some of the key computing innovations that have made gnomAD possible. But first, let me start with one number that expresses the hope and the promise of genomic science today. There are 7.8 billion people alive on the planet at this very moment. Statistically speaking, that means any mutation compatible with life probably exists in at least one human alive today. But whether a given genetic mutation has implications for health or disease is another question altogether. For an expectant couple or a patient with undiagnosed symptoms, this question could very well inform a life or death decision. Knowing this information would transform medicine as it's practiced today. If we could tell the effect of a genetic variant on a person's current and future health, not only could we provide more precise diagnoses to patients with genetic disease and help expecting parents make important decisions around family planning, we could also more accurately predict future medical complications for everyone, and we could propose new biological targets for drug development and therapeutics, paving the way for tomorrow's more effective medical treatments. Here at the Broad, we work with sequencing data from patients with rare, undiagnosed genetic diseases every day through our Center for Mendelian Genomics. The goal of the center is to identify the specific disease-causing variant for each patient, which can often help doctors to identify potential existing treatments. It also lays the scientific groundwork for developing new, more effective and targeted treatments. We recently received data from one patient, a 6-year-old boy with developmental delay and underdevelopment of the cerebellum, which is a region of the brain associated with balance and motor control. He was sent to us after standard clinical sequencing analysis of his family didn't reveal any causal mutations for his condition. So we went back to the drawing board to look for causal mutations that might have been overlooked in the initial analysis. But since any individual carries on average millions of genetic variants, how do we know which variants cause this patient's condition? This is a primary use case for a reference data set such as gnomAD. That is, in order to understand the impact of any one mutation, we have to put it in context. We want to be able to look at patterns of natural genetic variation that exist in a generally healthy adult population and to use them as background to evaluate variants we see in patients with disease. This was the key idea and the original vision behind the creation of gnomAD. Since 2012, a group of clinicians, population geneticists, computational biologists, and software engineers at the Broad Institute have been working together to build a data set that would allow people to draw insights from the vast amount of genetic variation encoded in living people today, and to help people like that 6-year-old boy and his family get some answers. When we started to build this data set, we identified 3 major tenets critical to the project's success. We needed enough samples to provide informative statistics. We needed an ancestrally diverse set of samples that represents the global population, and we needed to make the data free and open to all researchers. With respect to the first tenet, we were incredibly lucky to be at the Broad, which has been one of the world's largest producers of human sequencing data since its founding in 2004. And we were fortunate also to have formed scientific partnerships with researchers who really understood the scientific importance of sharing their data to advance genomic medicine. Over time, we've collected sequencing data from just under 200,000 unique individuals, which has allowed us to call over 644 million high-confidence short variants and about 455,000 structural variants. We're very careful to remove individuals with severe pediatric and Mendelian disease, and we remove all closely related individuals in our call set in order to ensure that the frequency estimates we provide for our variants in our call set are as representative of a general, healthy population as possible. We also put an enormous amount of effort into the quality control of our data so that researchers and clinicians are working with the most reliable, high-quality information we can provide. And I'll share more on how we use the cloud to run our pipelines shortly. The second tenet is not only critical to our scientific mission, but also to global health equity. Historically, genetics researchers have focused primarily on studying individuals of European ancestry, who represent only a narrow slice of existing global human diversity. By collecting and analyzing samples across a wider range of ancestry groups, we can discover new variants and better understand how variant frequencies can naturally differ across different populations around the world. gnomAD contains more genetic diversity than any other publicly available dataset of its size, and we're continuing to seek out collaborations and opportunities to incorporate samples from populations that have never been represented in gnomAD. None of these efforts would be of any benefit to the world, of course, if the data weren't freely available in accessible formats to the research community. To that end, we've devoted a great deal of thought and effort to the development and maintenance of a user-friendly, interactive web browser that allows non-experts to visualize and browse through the data. We also provide all the underlying data visualized in the web browser for local downloads, and with the help of the world's major cloud providers, including Amazon Web Services, Google Cloud, and Microsoft Azure, we host this data for free on the cloud, where anyone interested in this data can easily request compute resources to perform analyses on these data. Lastly, we make all our production and analysis code publicly available on GitHub. Everyone is welcome to clone our repository and to adapt it to use in their own research. Now, for the rest of today's talk, I want to share some more technical insights into how we've used the cloud to create this database and to make it available to the world. We run all our computational pipelines in the cloud. At the Broad, all raw sequencing data is processed and stored in the cloud, so we naturally pick up the Broad's output sequencing files, called CRAMs, from cloud storage buckets. When data comes to us from external collaborators, we upload those samples to a cloud storage bucket and then run the same processing pipeline used on internally sequenced Broad samples, so that we're applying uniform computational processing to all samples in gnomAD. This too runs entirely on the cloud. Our next step is to bring all the data from individual samples together into a single call set. Variant calls from each individual are stored in the genomic variant call format, also known as GVCF, and we gather all the GVCF file paths from the various cloud buckets in which they reside. Then we run a script that calls the Hail Python library to hierarchically merge individual GVCFs into multi-sample data matrices. Hail is a specialized Python library that's been built to handle large-scale genomics call sets, And Hale's hierarchical approach to creating call sets allows us to avoid recomputing previous merge steps for future call sets. So the final version of the call set today can itself be merged in a wholly modular way with other future multi-sample data matrices. This was what we did when we wanted to add a few thousand additional samples onto a previous call set for our latest genome release. Creating the v3.1 gnomAD release in the cloud in this manner cost us only about $400, when starting from scratch would have cost about $13,000. The entire process took only 6.5 wall clock hours and up to about 8,000 CPUs at a time to process roughly 40 terabytes of data. Once we have all the sequencing data together in a single call set, we next run our quality control pipelines on this data set. We divide quality control into two main stages. First, sample QC, when we identify low-quality samples that are affected by technical artifacts, as well as samples that are related to each other, and we remove them and their variant calls from downstream analyses. We also infer certain annotations, such as continental ancestry and sequencing platform, directly from the sequencing data, as these are annotations that are needed for rigorous sample QC. Next, we move to variant QC. We train several statistical models on both positive and negative variant training sets, which contain a mix of validated or true variants, as well as variants that look like technical artifacts, and apply these models to our call set to determine which variants look real in the database and which ones look like false calls. Both stages of QC are run as a series of scripts on the cloud, and as I mentioned earlier, you can find these scripts in the open source gnomAD QC GitHub repo at github.com/broadinstitute/gnomAD_QC. Data production at this scale is both expensive and compute intensive. At the peak of our compute usage for gnomAD production, we've requested up to 25,000 cores at once, and our recent call sets have cost well over $100,000 in compute to produce. So clearly, not every research institute or group can afford to produce and QC genomic call sets at this scale, which makes it all the more critical that we share the data we produce as widely as we can. This is where the cloud once again serves our goals as a project, by providing a natural platform and mechanism for access to researchers around the world. We currently host gnomAD as an open dataset on the world's 3 largest cloud providers, and all you need is an internet connection to download the data. For AWS users specifically, a number of tools already exist for working with gnomAD on AWS, and you can find them on the Registry of Open Data. Our ultimate goal is to put our data in the hands of the scientific community, and by doing so, empower researchers to find creative new ways to make use of the data. Since its launch in 2016, the gnomAD browser has served over 1.3 million unique users worldwide, with over 30 million lifetime views. gnomAD data is included in virtually every bioinformatics pipeline used in clinical diagnostics today and has contributed to the diagnosis of over 200,000 rare disease patients worldwide. At the Broad, the Center for Mendelian Genomics reports that gnomAD has been intrinsic in solving over 1,200 patient cases and in the discovery of 215 novel disease-causing genes to date. One of those novel disease genes turned out to be the causal gene for the 6-year-old boy I mentioned earlier. We found a variant in the patient's sequencing data that wasn't found in his parents' data and was completely absent from gnomAD. After reaching out to colleagues around the world, our collaborators found many additional patients with similar phenotypes and mutations in the same gene. So the diagnosis we made using gnomAD for this one patient became diagnoses for 15 other previously undiagnosed patients. This is just one example of how open genomic data is changing the world. We're enormously proud of the global impact gnomAD has achieved in its short history, but we know there's still a great deal of work to do to make this resource as useful as possible to the research community and to ensure that the benefits of that research flow back to all 7.8 billion people on the planet. To that end, we'd be delighted to hear your feedback on gnomAD and how we might improve it. And you can do so by filling out the user survey at broad.io/gnomad_user_survey. You can also reach us with general comments, suggestions, bug reports, and questions at our email address, gnomad@broadinstitute.org. We're really looking forward to hearing about the insights and breakthroughs that you, the AWS Healthcare and Life Sciences community, make in the future with gnomAD data, so please be in touch. And thank you all so much for joining Aaron and me to hear about how open genomic data from the gnomAD project is powering some of the most impactful science being done in the world today.